The sex chromosomes that are present in humans, and most animals for that matter, are going to be the X chromosome and the Y chromosome. And these two chromosomes, as I've drawn them here on this slide, notice that they don't look alike. So these are what we would call non-homologous chromosomes. And since they're non-homologous, they are going to contain different genes, or you could say different loci. So they don't have the same thing on them. This is going to affect the inheritance patterns if we're talking about genes that are located on the X chromosome or something that's located on the Y chromosome. So anytime we talk about genes on these chromosomes, we do refer to them as sex-linked genes because they are located actually on the sex chromosomes. And we do have to understand that the number of copies of these is going to be different depending on whether we're talking about a female or a male. So for females are going to have 2 X chromosomes, but males are going to get 1 X and 1 Y. So if we're talking about something that's located on the X chromosome, for a male would only get 1 copy of that because they only have 1 X chromosome. But a female, she is going to have 2 X chromosomes, and so she will have 2 copies of that gene, and those 2 copies of that gene may be the same or they could be different alleles from each other. So if we talk about the female situation first in a little more detail, the female has 2 X's there. And if we have a gene that has 2 alleles present, we have a capital and a lowercase, so we have a dominant and a recessive, we could have a female that has 2 copies of the big A, and I'm going to write it right there on the X so that we can keep in mind that this is a gene that's located on the X chromosome. So that right there would be what we would refer to as homozygous dominant, because she has 2 copies of the dominant allele, and she would then have the big A phenotype, whatever that happened to be. It is also possible to have a female that is heterozygous, so she gets 1 copy of each, and if the big A is dominant, then she would look like big A. And then it's also possible for her to have the lowercase version, in which case she would, 2 copies of it, she would be homozygous recessive. Now this is a little different from the situation that we have in males. So if we talked about the male and what the difference is going to be for the male, with the male, if I just draw it in a different color here, the male gets 1 X and 1 Y. So he can either get a big A version or he can get a little a version. And those are really the 2 options. So notice that it's not possible for the male to be heterozygous, because to be heterozygous, you would have to have 2 present. So one word that we do use to refer to the male genotypes when we're talking about genes that are located on the X chromosome is we call males hemizygous. And that term hemizygous is again just referring to the fact that there is only 1 allele there. So if the male has the capital A version, he is going to look dominant, and if he has the lowercase version, he's going to look recessive. So let's take a look at a cross and use a Punnett square where we're talking about genes that are located on the X chromosome. So the scenario we're going to look at is going to involve color blindness. And there are multiple types of color blindness, but for the example that we're going to look at, it's really red-green color blindness. This is located on the X chromosome. So that's where the gene is at, and there's 2 alleles for it. So I've got a capital B that's going to represent the normal version. So that would be someone that is going to be able to see color just fine. And then we have the recessive version, which is a lowercase b, and that represents someone that is going to be colorblind. So suppose that a heterozygous female and a colorblind male have a child. And what we want to know is what is the probability that the child is colorblind, and I'm just going to write it like that, CB. So the first step with something like this is to write out what the genotypes of the 2 parents are. So we know that we have a heterozygous female. So when we write her genotype, we want to go ahead and put 2 X's there so that we remember that she has 2 X's, and then she's heterozygous. So she's going to get a capital B and a lowercase b. Notice that that would be her genotype. If we wanted to say her phenotype, she is actually going to be normal. So she's going to be able to see color. If she was being crossed with a male, we want to go ahead and write an X and a Y for him, and he is colorblind. And remember that there's only 2 options when we talk about the males. They either get the capital version or they get the lowercase version. So that makes it a little easier to tell their genotypes. He's going to get the lowercase b because he is colorblind. So this is the cross that we are making. So if we draw our Punnett square over here, we're going to do it very similar to what we did before with the monohybrid crosses, in that the female, these 2 X's are going to separate during meiosis. So if we were to put her eggs, possible eggs, down the side here, half the time she's going to contribute the X with the big B, and half the time she's going to contribute the one with the little b. And then from the male, half the time he contributes his X, and half the time he contributes his Y. So this is our square. And now if we just go through and we fill in the boxes, we fill them out the same way. We look above and to the side to figure out what we are going to write in the squares. And now this is what we get. So when we look at this particular square, notice that we have, if we assign the phenotypes, this one is normal. Okay, they can see just fine because they have the capital B. This one is normal. And then both of these bottom boxes are actually going to be colorblind because they don't have a capital B there. When we look at these boxes, not only can we figure out whether they're going to be colorblind or not, but we can also see whether they're going to be female children or male children. So if we were just to answer the question, the probability the child is colorblind, the bottom 2 boxes are 2 out of 4, or 1/2 are going to actually be colorblind. We could also be a little more specific with this, and we could say, what is the probability that if they had a male child, that he would be colorblind? In this case, it's still going to be 1/2, but what we would specifically do is we would look at the male children, which would be these 2 boxes here, and we'd say 1 out of 2 are going to be colorblind if they had a male. And we could do the same thing with the females. We could say, what is the probability that if they have a female, and now we just want to look at the females, what is the probability that she will be colorblind? And in this example, it does turn out that it's 50% as well, or half. It is possible when you do the crosses that you're going to have different probabilities for the male children versus the female children. Just in this particular scenario, that didn't happen.